Okay, let's get started. Okay, so second talk. This talk is, unlike the first one, we're going to talk about learning in a different context. We're going to talk about it in terms of motor movements, motor control. So the common theme here is how to help people. So in this case, I'm going to show you basically why movements are important and what happens when people have deficits in movement. And it turns out some of the same themes I was talking about also carry over, because it turns out that the reason that people have problems with the movements, it's also related to brain circuit. It's a very specific brain circuit, in fact, how this happens. It's even more specific than what we know from the previous lecture, which is more of a cognitive type of effect, emotional cognitive effect. So the reason I call it playing the piano without the brain is because this next structure that I'm going to talk about is a location that most people think is not even part of kind of the higher functional brain. It's this area called the cerebellum, which is this lower part of the brain, which is down here. And that area of the brain is very well conserved across different species of animals. So it's been around for a long, long time in terms of evolution. And it also has the most number of cells in the human brain that we know of, in the form of these cells called granule cells. The question is, why do we have so many cells there? What do they do? And what circuits do they do that allow us to have fine-tuned movements? And so, yeah, let's get started. So we already talked about this, but I just want to emphasize once again that I'm linking the social global aspects of behavior and humans in general down to the human brain, and in this case even closer down to subcompartments of the cell. So it turns out there's just as many, almost as many synapses on a single cell in this case as there are a number of cells in the cerebellum. And so the question is what all those synapses are doing. So again, we're going to link the behavior and the social level down to this very microscopic level where the circuits are that we're actually going to study. Okay, so why are movements important? So if you think about it, same thing as what we talked about even in the last lecture and everything else in between. Everything consists of movements, actually. If the animal was moving around in the cage and they were listening and they were freezing, that was all movements. Everything was controlling of the muscles. So if you think about it, that's one of the most important things in what the human brain can do. It can sense external perception, and then it can also use that information to move in the real world. And if you can't move, then you can't do anything. Right? So turns out cerebellum plays a huge part in the motor control, which is control of these movements. It has a large number of cells in the human brain, as I was saying. And then there's a bunch of different cerebral diseases, related diseases, in which people have lesions of different subparts of the cerebellum, and they see inability in each of these different areas. So I'm going to talk about that in a second, so that to illustrate why it's important to study this area of the brain and this topic in general. So one is called hypotonia. So that is basically a reduced resistance of your muscle tone, right? So one of the most common ways people look at this is, have you guys tried to basically swing your arm like a pendulum? So most of the time you can just do it, right? So if somebody starts you here, you can actually swing it like a pendulum, right? So eventually you kind of, have this resistance decrease, right? So it turns out there are some people who doesn't have that kind of reduced resistance, right? So this resistance that the muscle have. So instead of like kind of controlling and kind of rapidly decreasing, they actually just swing nonstop without ability to change the tone of their muscle. There's also a phenomenon that's called Astacio. So Astacio, basically, you might have even seen people on the street that do this, but basically they can't really walk straight. So you might have seen, maybe homeless people or something, or maybe just older people, they walk like this, right? Have you seen this? So that is actually the inability to walk sort of appropriately. They actually cannot control, what I do is just, they cannot control the right side of their body, which indicates a left side deficit in their cerebellum, right? And it's because of this deficit in the ability to control certain motor movements on the contralateral side of the brain. And so they spread their feet like this to kind of help them maintain sort of this balance, but inability, they cannot really control that side well in a fine-tuned way. There's also ataxia, which is the irregular rhythmic movements. So for example, they can't do things like this, right? Flipping the hands. So I know for us it's just like, why wouldn't you be able to do it? It seems like it's very easy, but if you have cerebral damage, you don't have the ability to kind of, one, have this sort of fine-tuned movements, sort of stuff like this, okay, like point somewhere and then point to your nose. They can't do this very well, and if they do, they have these tremor. And the other thing is that even if they're able to do it, they can't control exactly where it goes. Okay, so what I was just saying is intention tremor. That is basically this tremor that you can see when they kind of. So actually, it's probably easier to show you the movie. This is intention tremor. Let me show you first. So he's got his hand right there, and he can't control it from tremoring. So you see that? He can't control this. He tries not to do this. So one thing you notice is that on his way over, right? You notice that on his way, so when he's trying to point, he can't point exactly. But if you notice on his way over, I'll show you again, he actually has no problems. So this time concentrate on the part where he's actually moving. Okay, watch the part where he's actually making the movements, okay? So this part is okay, and this part is tremoring, and then now let's see, he's going to make a movement right now. You see that? Perfect. No tremor. See, I'll show you, we see again, okay? So watch when he points back to the place. No problems. This is a deficit specifically at a stationary posture. Once he has the motor program to go from this spot to this part, he has no problem when he's actually doing the movement. It's when he's pointing he's having this tremor, right? So this is kind of very typical of these cerebellar deficits that we want to help people to overcome. And it's the fact that it's a very specific thing, just like here, very specific thing. There's another thing that a lot of people see is like when you put your knee up and then when you do this, right? Right. I mean, I'm just acting right now, but you get this knee up, right? There are certain people that just doesn't do it. So things like this where it's very specific to a certain part of the body or certain, fine-tuned movements, it kind of indicates cerebellar problems, right? Okay, so, sorry, it's a little bit cut off, isn't it? So what is it about the structure of the cerebellum that allows it to have these fine-tuned movements? Because, clearly when we have damage in the cerebellum, in different areas of the cerebellum, you have these very specific deficits, right? So what circuits in the cerebellum is actually doing this, right? So what we know first of all is that the cerebellum is very compartmentalized, right? Again, we don't have to know all this information. This is kind of just illustrate this idea that cerebellum is not just one big compartment. It's a lot of specific compartments. So first of all, there's these things called the deep nuclei, right? So the deep nuclei actually has to do with some volitional movements. So because what we know is that the motor cortex sends input there, and when you have lesion in that area, actually you almost cannot make movements. It's a very deep, very basic deficit when you get the lesion there, right? It turns out it also has vestibular input. What that means is that, for example, it's able to control eye tracking, so the pursuit of something in the visual space using your eyes, when you're looking around, that's actually cerebellum dependent, and that's based on the input that it receives from the vestibular system. It also has input coming up from the spinal cord, right, and the red nucleus. And so one of the things that people talk about in terms of that input is that cerebellum seems to be getting a copy of the motor program. Okay? It sounds complicated, but what it means is the following. When your body makes a movement, right, it has a volitional will to make some movement. We think that that could be generated in the cortex, for example, secondary motor cortex, or basal ganglia, But when that volitional action happens, it turns out the cerebellum gets sort of a copy of the command. Okay? The reason people know is they basically study this by doing inactivations and things like that. But that secondary command is actually what's allowing the cerebellum to fine-tune the movements. So imagine that some other part of the brain, okay, is kind of giving the brain, or giving the human being a motor program. Like say, I want to move from here, here to here, just like that patient earlier. Now, what's happening is that volitional movement, actually information gets to the cerebellum. Cerebellum is not necessarily the thing that's allowing you to say go there, like just go there. But the cerebellum is allowing you to go there in the correct way. If it has a copy of the information, then as this movement happens, the cerebellum can fix it. You see what I'm saying? So the fine-tuning occurs in the cerebellum. It's not necessarily the thing that's allowing you to make the movement, but it's definitely the thing that's telling you how to do it. Okay, so it's a very subtle, that's why I was kind of using this piano metaphor, right? Because it's a very fine-tuning of skills. Yeah. Another example I like to use is a pitcher throwing a baseball, right? So if you have a lesion in the cerebellar cortex, someone might still be able to throw a ball, right, in a very crude way. But there's no way he's going to be able to throw it like pinpoint right at the strike zone, the way you do with the cerebellum. Because the cerebellum fine-tunes that output as the pitcher is making the pitch. Go a little bit more this way, a little bit less this way, a little bit more this way, a little bit more less this way, all throughout this movement, so that you're able to make a very specific, fine-tuned gesture. Okay? But I think the thing that we have to realize is that this gesture happens every day, everywhere, every single movement that you're making. Because, like me looking around the room, that is a very fine gesture telling my eyes to move here and then here and then here. Everything that you do almost is a very specifically well-tuned thing. So it's hard to imagine what would happen when you lose your cerebellum. And so some of those diseases kind of illustrate what would happen. So, so what we're going to concentrate on this class actually is the lateral cerebellum, the cerebellar cortex, basically. That should probably be stated more correctly. It's cerebellar cortex. That's getting the input from the cortical areas. So presumably somebody's telling them, what we're doing, what command we're making, through the pons into the cerebellar cortex. And the cerebellar cortex actually goes back and innervates the dentate and also the prefrontal cortex. So it's kind of a loop. It goes through the cerebellum, and then the cerebellum provides information back. And it turns out the cerebellum has a very specific output cell structure, so cell type, that allows this to happen. And when you fine-tune the way these output cells are performing, you actually can get very specific movements, very fine-tuned movements. Okay. So I hope it's, for me it's very fascinating. Hopefully you get some sense of the mystery and curiosity of thinking, well, we have these very fine-tuned movements every day, but how do we actually make that specific thing? It seems very obvious to us because, we just walk around and move around this real world, but it turns out that all those tiny little processes are very critical and very, very well fine-tuned. Okay. All right, so having said that, I hope you understand how important cerebellum is. So now I'm going to talk about some studies that led us to this organization of the cerebellar cortex. Okay. I'll talk about the nuclei, deep nuclei a little bit as well, but those are kind of the only two main structures that I'm going to emphasize. It's the cortex of the cerebellum versus the deep nucleus in the cerebellum. Okay. So I think we can ignore the one on the left because it's just a little bit more detailed than I like. But what we want to look at first is this stuff. Let's start with the one on the right, I think. Oh, actually, let's start with the one in the middle, the gross organization, and then I'll move on to the right. So the first thing we can do is look at the cerebral cortex in terms of the type of cells that are there. One way to organize them. The other way is to look at the input and output in the circuit that's actually there. So first, let's look at the layers, right? So first you have the granule cell layers. So the granule cell layer actually gets some input from the sensory areas. It turns out there's a lot of granule cells, right? So this is the most numerous cell type in the human body, right? Or sorry, the most numerous neuron in the brain, right? So why is it that we have so many cell types? These are all very small cells, actually. And it turns out most of them are not active most of the time. So only a very small number of them are active at any time. So it's kind of a mystery, but I'll tell you about some of the theories we have about these cell types later. It turns out there's also interneurons in there. So interneuron just means that these are inhibitory cells. So granule cells are excitatory cells. You give them an input, and then they excite whoever is downstream from them, the next guy over. Okay? The interneurons are inhibitory, which means when you excite them, they release inhibitory transmitters, which inhibit the cells that are around them. So in this case, inhibiting mostly the granule cells. So the Golgi cells are much bigger. So you have an input coming in that's into both the inhibitory and the excitatory cells, actually. And the question is how that information propagates forward. The other main layer is the output. So I talked about granule cell receiving some input. Now the other one is the Purkinje cells. So the Purkinje cell is really your basic output of the cortex. Okay? It is the thing that after all this information kind of integrates and comes together and gets processed in certain ways, the Purkinje cells are the ones that are giving you the output. Very characteristic cells. I'll show you a picture later. Huge cells with very big dendrites that look like trees. Okay? They're very big, 70 microns each, and they are the ones that actually project to the deep nuclei. So I talked about deep nuclei before. This is the cerebral cortex. The cortex output are the Purkinje cells. It turns out, though, interestingly, that the Purkinje cells, even though they're output of this cortex system, cerebellar cortex system, they're actually inhibitory. So, so there's a lot of intricacies in the brain, right? But one of them is this, right? The cerebellar cortex outputs are actually inhibitory. It's kind of tricky. So what that means is that whatever the nuclei is doing, it's modifying it in such a way to inhibit them. Again, it kind of makes sense because if you think about it, the cerebellar cortex, again, is fine-tuning. It's not the thing that's willing things to happen. So in that case, what do you want? if it's a fine-tuning system, first you want to be able to process a lot of information. So maybe granule cells, because you have a lot of them, right? The other thing is that you want to modify existing information. How do you do that? You inhibit outputs of other cell types. So the reason I find the cerebral cortex very exciting is kind of these two properties, right? Because it's not very simple structures, in the brain that says, okay, let's just do this. We come up with a program to do this. It's actually the thing that's kind of fine-tuning in the background. It's like, oh, let's change this, let's change this. Very kind of mathematically calculated, how do you throw this ball right at the strike zone and not outside the strike zone? That kind of fine-tuning. So it's very fascinating to study the structure and how computationally it's actually doing this. The last layer I'll talk about is the molecular layer. The molecular layer, we already talked about kind of the input and the output. The molecular layer is the guys that's relaying the information. So molecular layer, the reason it's called molecular layer is because there's a lot of fibers there. Okay? Fiber just means they're like axons of cells. So these are like the thin fibers, right? So these fibers are actually the output axons of the granule cells. So I'm sorry that I know some of you haven't had sort of, proper neuroscience, but but basically a neuron has input coming into their synapses, right? Their dendrites, and then their output are most of the time on the axons. So the axons are a little bit longer branch that goes out and innervate other cells. So the input comes in the synapse, output goes out in the fibers. The fibers in this case are called parallel fibers, but they are actually just the axons of the granule cells. So this is the same cell type as the granule cell layer. I know there's a lot of information here, but don't worry, I'm going to say it again, like 3 more times. Just in a different way, okay? In the molecular layer, in addition to this, there's actually also stellate and basket cells, but those are just interneurons, so you don't have to worry about too much. They're just inhibitory cell types. Right? They're again modifying the input and stuff. It turns out these parallel fibers actually innervate Purkinje cells. So the circuit is actually very simple. So now you can look at the diagram to the right. So this input to the cerebellum comes in these 2 fiber types called mossy fibers and climbing fibers, which I will talk about later. But right now, what you know is in the cortex, once the information goes to the granule cells, so the guys on the right, I hope you guys can see. I know it's hard to see here. But once it gets into the granule cells, in the granule cell there, they propagate up, and these are just their axons. They're just really long, right? Up to several millimeter long, which in the brain is unheard of. Long, long, long. So, but these parallel fibers are just the axons of these granule cells, right? They're actually not myelinated, which means you have slow conduction speeds. But what they do is actually innervate these Purkinje cells. So Purkinje cells, each of the dendrites actually is innervated by these axons, right? And the output of the Purkinje cell goes to the deep nuclei. Right. So it's actually modifying the way that the deep nuclei is fired. Sorry, I'm losing my voice. So I promised to show you a picture of the Purkinje cell, and this is it. So again, this is the output layer of the cerebellum. Right? These are actually inhibitory, like I was saying. Very beautiful cells. So each of these is, so the red is actually stating for the nucleus of the cell, nuclei of the cell. And then what you can see, these branches on the top there, that's the dendrite. Dendrite means it's the thing that's receiving the information. It's where the synapses are. But you notice these dendrites are, I mean, it's just humongous, huge, right? It's like a tree, like a forest. So when you put a dye in each individual Purkinje cell, you can see these little branches, right? Incredibly dramatic cells. So one of the inputs of the cerebellum in the cerebellum actually goes directly to the output, which is the Purkinje cell. And these are the climbing fibers, which I'll talk about later. but those actually go one to one into these Purkinje cells. So in other words, one climbing fiber goes into one Purkinje cell and wrap around their dendrites. I will talk about that soon. Okay, so I already told you the story, but now I'm going to repeat the story so that you remember it, okay? It's the same information. Just going to say it in a different way. Okay, so first of all, parallel fibers are the middleman, right? So they're the people who are the axons of granule cells, which project to the Purkinje cell, and the Purkinje cells are the output. Okay, so this is the diagram I'll show you. Now I want to elaborate on the input and outputs a little bit more, right? Okay. So first, let's talk about the two inputs. I told you that there's two sort of extra inputs into the cerebellar cortex, and these are the two, right? So one input, let's first concentrate on this lower one down here, mossy fiber. Okay. So this input is basically giving you the information in your body, basically sensory input, right? Posture, tone, anything you can think of, right? Motor stuff from the motor cortices, stuff from the lower body, right? And that input is basically motor, right? So it's giving you motor information. So it's the thing that is kind of the information that the cerebellum is eventually going to be modifying, right? And also kind of processing, right? So first of all, there's a lot of convergence in the system. What that means, what convergence means is that there's a lot of input coming into the system, and then because these mossy fibers, I didn't know you could do that. Okay. Because these mossy fiber actually does the following. Each one of these branches actually goes into this structure. You don't have to necessarily know, but it's called the glomerulus. But this structure actually has like 5 granule cells around it. So what that means is that this information comes in, and you have different mossy fiber coming in to innervate one, to each granule cell is getting input from different mossy fibers. That's the simplest way I can say it. So what that means is convergence of information going into a single granule cell. Okay. Now each mossy fiber also does more than one, synapse there. But overall the convergence is, overall the processing is convergent. In other words, even though there's so many granule cells, each one of them is already getting information from multiple sources already. Right? So why would you want to have convergence? Well, one possibility is that you have so much stuff in the brain, right? I mean, technically, you're getting information from every skin cell on your body, right? In addition to the muscle tone and the internal, that is a shitload of information, okay? So even though you have a shitload of granule cells, that information is still selected already, right? In terms of what you can actually pay attention to in terms of the motor system. So this is one reason possibly why you have convergence, even going into the input. Right? Everybody knows now what convergence means? It's okay, more or less. Okay. All right. And then the other thing is that in this convergent input, the activity that's coming in is actually simple spikes, which just means they just fire. Just go boom, 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 right? A little bit more quickly than what I'm indicating. But just your regular simple spikes. In contrary to what I'm going to talk about next, which are the climbing fiber inputs, right? So climbing fiber input is completely different from the mossy fiber input. The climbing fiber input, first of all, let me describe what it is. It's a one-to-one connection between the climbing fiber and the Purkinje cell, right? Okay. So the climbing fiber, this guy basically, from the inferior olive coming here. Now the inferior olive actually gets information from the deep nuclei, so it's actually a loop. Okay. Now, what people usually think of these guys is they think of it as an error-correcting code. Okay. I'll show you what that means in a second. So first of all,